Hi everyone, welcome to today's webinar. I think we're just going to wait about a minute or so for everyone to join, and then we'll go ahead and get started. Hope everyone's having a good morning or afternoon or evening, depending where you're located. Daniel and I are 7 hours apart.
Yep. For me, it's already evening, 7 o'clock in the evening.
And it's noon here on the eastern side of the US. All right, so we're about a minute in, so let's go ahead and get started. Hi, everyone, and welcome to today's webinar, From FAIR Data to FAIR Analytics. I'm Alicia, a product manager here at CodeOcean, and I'm joined today by Daniel Koster, our VP of Product. So before we get started, just a few housekeeping items. First, this session is being recorded, and we'll send you all the recording after the event. Second, we'd love to get some questions from the audience today, so please feel free to submit questions at any time throughout the webinar using the chat on the right-hand side, and we'll answer them at the end of the presentation. So I'll quickly introduce myself and my background. I did my doctorate in molecular and cell biology at the University of Connecticut. My research focus was on epigenetics and genomic imprinting, and I've since worked with many researchers in biotech and biopharma in various genomics service lines.
Hi, I'm Daniel Koster. I'm VP Product at CodeOcean. I did my PhD in single molecule biophysics in Delft University in Holland. And my postdoc I did at the Weizmann Institute in single cell genomics. And then I was a team lead of a bioinformatician bioinformatics team in Israel. And we developed some software for the phenotyping of plant using deep learning, as well as GWAS studies of plant populations.
All right. Now, today we plan to cover some key differences between FAIR data and FAIR analytics, some actionable guidance to overcome challenges in making experiments fair and reproducible. And lastly, we will cover some real-world case studies and examples of FAIR analytics implementation in the life sciences. So with that, let's dive in and start at the beginning with FAIR data. Now, I hope most of us in the audience are here because there is interest in at least some aspect of FAIR. The ultimate goal of FAIR is to optimize for the reuse of data. But we think here at CodeOcean that FAIR data is not enough. Being able to simply reuse data is not enough. So we invite you all to consider the entire experiment. So the entire process that uses data to generate results and gain insights from them. Data being fair is not good enough if the result that comes from it is not reproducible. And this is also where the second R in FAIR analytics comes into play. So this R is for reproducible. And this is really where we find that things are lacking, and that's exactly what we're going to be discussing today.
So let's dive in a little bit more using on this concept of reproducibility using an analogy. And the analogy we chosen was the taste of beer. So some companies such as Heineken are proud to be able to reproduce the taste of beer no matter where it was created, was brewed. And so basically there are three components to that process. So there is the ingredients, such as yeast, barley, and water, and of course there are many more. And there's the recipe, basically the instructions for how to treat those ingredients. And then there's the equipment, right, which is basically the apparatus to work on these ingredients, such as barrels or the combine to harvest the barley, and the reverse osmosis for the water, for example. And so to be able to reproduce the taste of beer, you have to, of course, regulate very tightly the ingredients that you're using, right? And in the analogy of a computational experiment, that would be, for example, the data, right? And then you also have to tightly regulate the recipe, very precise in exactly what mineral composition you need, what yeast to use, quality control, the sequence, and all of that. And that would be, of course, the code. And then the equipment is exactly the kind of barrel that you use, the kind of combine, the kind of filter system that you use. And that would be, in our analogy, the compute resources, right? The compute platform. So to reproduce the taste, you don't only have to be very precise about the ingredients, the recipe, and the equipment. You also have to make sure that they're all three together, very tightly regulated to create the result, which in this case is the taste. So that is the analogy that we're going with today. So the first ingredient for a computational experiment is, of course, the data.
Right. So, and going off of this analogy, ideally the data that we're using should be findable, accessible, interoperable, and reusable. Now, these FAIR principles were defined less than a decade ago, and they are really becoming more and more relevant as the amount of data being generated every year continues to grow exponentially. These FAIR principles also emphasize the capacity of the computational systems that generate them and end up using that data. And fairly recently, in 2022, they have been described as a must so that data mining and AI can optimize extraction of scientific information and insights from the data. And as I mentioned before, fair data alone is clearly not enough. So, going back to the earlier analogy, having the right strain of yeast is not enough to get the same exact beer every single time. And to this day, in the scientific community, we are still facing the ongoing reproducibility crisis, in which many scientific studies are difficult to, or, in some cases, just plain impossible to reproduce. And the cost of this is really quite staggering. We're looking at an estimated $28 billion a year is spent on non-reproducible preclinical research, and that's in the USA alone. It's even worse when you consider that new research constantly builds on previous findings. And how can you trust the credibility of a result that comes from an experiment or is based on another experiment that is not reproducible? So reproducibility crisis, it really affects all fields of research across the board. And as recently as just a couple weeks ago, yet another study was published, this time looking at over 27,000 Jupyter notebooks from over 3,000 publications. These are published results. So now we invite you all to take a quick moment to answer this poll that I believe is going to pop up on your screens. The question is, how many of these Jupyter notebooks do you think they were able to successfully reproduce in this study? So I hope everyone can see this on their screens. We'll go ahead and give everyone about 30 seconds to submit an answer. And just an interesting point to be made here, Jupyter Notebooks are a very popular way to do bioinformatics and data science work today. And some might think that because, it helps you organize and document your code and your progress along with results, kind of sounds like reproducing a notebook shouldn't be that hard, right? So I guess we'll find out. Let's give everyone just a few more seconds. And I hope answers are in. I can't see on my side, but I think we can now go ahead and close the poll. Thank you all for your participation, and let's see if we have some results. I do not see them.
Yeah, so I see the results. So there's 3% of the participants thought it was 45%, then 16% was 23%, 45% thought it was 11.7% reproducible, and 35% thought it was 3.2% reproducible, which means that the winner is 11.7%. And we are here to say that the correct answer for this poll is 3.2% only. That is a result of this study.
Thank you, Daniel. So, yeah, only 3.2%, a very, very small number of the notebooks in this study could be successfully reproduced. Most of them actually could not be executed at all. And they mentioned it was mostly due to issues with dependencies. So I think we can all agree that fair data is not enough. It certainly does not get us to computational reproducibility.
Right. So we share these very low numbers. Oh, sorry. Go ahead.
Yeah, anyway, as scientists and researchers, I think most of us, if not all, have encountered a result that isn't reproducible, either our own result or another scientist's result. Myself personally, working as a grad student in a lab, on multiple occasions, my PI asked me to reproduce a finding from a previous lab member who was no longer around. And usually, what that looks like, it's asking around to see where the data had been moved to. How do I install all of the packages needed to run the script? Am I even looking at the right version of the script? It was never an easy, straightforward process. And of course, the person to answer all of my questions was no longer around, or they just couldn't remember what they did. And a point that we want to make is that this is certainly not, a unique issue that I came across. We do believe that this is a global issue across the board.
Right. So indeed, I think that's really true, Alicia. I think most people might be tempted to think that, if they have a problem reproducing their own data in some company, that it might be because of them or because of the company, or maybe if it was the same in the previous company they work with, they have this idea that maybe it's their fault, or in their specific company. But like you said, it's not a local problem. It's a global problem across the whole domain. And we at Corrosion, we see all of those companies, tens of companies that we get to know very intimately, and we find that this is very common, right? And the second thing is we see patterns, right, that we can share here in this webinar, which I think which is great. So again, if we go to those three components of a reproducible experiment, the code, the data, and then the environment, you can see that all of those three have their own issues, right? And this is just a very partial list, right? The list is very, very long. But for example, the code, right? The first thing you have to find out is where the code is that generated that result that you want to reproduce. So finding it is hard. Is it in a repo? What's the repo's name? If you find it, do I have access? If I don't have access, then who am I going to ask? Right? And then if you're lucky to find it, right, then sometimes there's no README file or no documentation, so you don't know how to run it. And same goes for data. Like, where is the data? Where is it stored? Is it what S3 bucket, what drive? Maybe it's stored on some local computer. Who owns it? Do I have access? If I don't, who to ask? And then very often the data itself is sort of not the exact data that you need for the experiment. There's still some pre-processing that you have to do. And then that is not always documented very well. So it's unclear what the actual data is you should use. And if there are multiple versions, then what version should I use? And then we have the environment, right? If you're trying to reproduce an experiment of, let's say, 2 years ago or 3 years ago, let alone 10 years ago, the chances that that exact environment that generated that specific result still exists is almost zero, right? So the server has been torn down, it's upgraded, and then you find yourself rebuilding the same environment again that should run that experiment. And first of all, you have to figure out what that environment is, and then you have the issue of installing the packages in the right order. There might be conflicts, there might be versions that are not compatible, and then there might be conflicts with the operating system, and then we have the compute resources. Is there a GPU? What kind of GPU? How much memory do I use? It's not always clear what the answer there is. And so this is just a very, very partial list of the problems you might encounter. But ultimately, we think that these kind of quote unquote mundane problems are exactly the source of this problem that we just described in terms of lack of reproducibility. So that is really only just one experiment. So now imagine you have like a pipeline of experiments. We know that in bioinformatics often we have pipelines. Sometimes um, they're run in the same environment, but very often they're run in different kind of environments. So for my personal experience, I can say that in my previous job, I ran a project with over 7 academic groups and 4 industry research groups that all sort of rallied together around a common theme, which is to calculate some phenotype, some appearance of fruits, right? So in this specific case, it was the shininess of a tomato. And so we had various parts of that process. One was detecting the tomato, then it was finding out whether there was any occlusion of leaves and stems, then the orientation of the tomato, then the measurement of the size of the tomato in centimeters, right? So which uses a 3D camera, and then there was the shine. These were all neural networks that were developed in different labs, and it sort of worked individually for in that lab, but when we tried to stitch all of those together, it was a nightmare because exactly because of the reasons we just mentioned, right? The environment was different. The data wasn't clear where it was lack of permission sometimes, so it was really really hard to stitch all of them together. So this is really compounding the problems that we just mentioned at the level of the individual experiment, right?
Yeah, and just to emphasize that this siloed approach that Daniel just described, where multiple teams or even multiple individuals are participating in a workflow, but they're all kind of doing their own thing. They're using their own tools, their own analyses, and then you have to kind of stitch everything together to reach that final result. This siloed approach, we're seeing it. It's really a universal concept. Every company, every customer that we've talked to, it's really something that we're seeing a lot of, and it is, we believe, part of the problem.
Right. So the way to solve this, right, and this is one of the main messages of our webinar, is that if you have results in the organization, for example, in this slide, file two, that is the result of project one, right, and you want to reproduce that specific file, then what you have to do is link that file, that result, to individual files in the code repository that you might have, like code, data, and also the environment. So all of those three have to be linked directly to that individual file. Now, in a different project, you would have, for example, another file and may have a different path, right, in this graph. Different input files, different code, different environments. But the point is you have to link all of them together. And then, of course, you can also have versions, different versions as you iterate through your project. You might have version 1, 2, and 3 of file 4 as a result, which may be linked to different versions of the code as you iterate the code and the data and the Docker environment, for example. Now, what is interesting here is that on the one hand, we see this huge crisis that we find in reproducibility, but here we are saying that the code and the data and the environment is the key concepts you have to regulate, but there's technology in place to manage those aspects. So we have Git, for example, for code. We have S3 or databases for data. And then we have, for example, Docker to manage to encode the environment, if you will. So now you might ask, so if we have all of these technologies already in place, why is it so hard to reproduce data, right? And the answer is that actually integrating those technologies for users that are not sort of expert computer scientists with a sort of an engineering background, that is extremely hard to do, right? So the user experience and the whole abstraction level that you have to build in order for for scientists that are not computer experts to use this effectively, that is extremely hard, and that is basically the solution of this problem, right? And then what we do at CodeOcean, for example, for instance, is take that path, this slice through the database, so all of the essential files from the triplet, the code, data, and environment, merge it together with your data, and then package it into one entity. So at CodeOcean, we call that a capsule, right? You can call it anything you want, of course. And now the thing is, if you make the compute resources available to that entity, right, then you can run the entire thing from the input through the compute resources and obtain the result every time, because you have everything you need to reproduce that result anytime. So that makes it reproducible. And then because it is also a standalone entity, you can also transfer it just like a YouTube link to someone else and really enhance collaboration. And that is pretty great about that concept, right?
Yeah, and I think the essential triplet also really highlights why fair data is not enough. All three of these core components must be managed and in the context of the specific experiment with the associated result that you get with it. So when these objects are stored together as a unit, every time you rerun an experiment, you're going to get the exact same result every time. And that is a big part of FAIR analytics. There we go. And beyond the essential triplet, these experiments, this slice of that database, they also need to be associated with the appropriate metadata, and they must follow commonly agreed upon standards to be fair. Because without these, even if a user has access to the essential triplet, even if they found it, if the metadata isn't there, if it doesn't follow a standard, they still need to know how to run and rerun the experiment successfully. So I mentioned at the beginning of this presentation that the ultimate goal of FAIR is to optimize for the reuse of data. With FAIRR Analytics, we are optimizing for the reuse of the entire computational experiment. Now, we'd like to present a few case studies that demonstrate what FAIR analytics looks like in practice. So this first one, this is a case where one senior scientist created a workflow that was used, reused, and built upon within their team and organization. So this workflow that they created serves as a trusted standard. And we were told that this customer saw increased productivity by up to 30% from this. It's because other users, other scientists within the organization were able to take this workflow, reuse it, build upon it easily without needing to put all of those pieces together. And the real value to this is that a single expert in the domain can set the standard of how things are done, and that has an impact throughout the entire organization. And another case study that we saw as part of due diligence prior to a legal disclosure, one of our customers received new clinical trial data and they were asked to rerun their complex analytical pipeline using this new data with a 12-hour deadline. So they had to present results 12 hours after receiving this new data. Long story short, they were successful, and they told us it was because they had CodeOcean, because they had everything tracked and ready to go for this pipeline of theirs. And previously, this would have been unheard of, but because of our system and how it implements FAIR Analytics, there was a happy ending to the story.
And the third case study is from the Allen Institute, which is a great nonprofit research institute in Washington State. And they created a quite complex pipeline for the analysis of calcium channels from brains of mice. And so they had many advantages when using CodeOcean, first of which was the creation of the pipeline in a very easy manner, in the sense that scientists themselves could actually create the pipeline and weren't reliant on a whole team of engineers that are often very overbooked. That is one. And the second thing is that once that pipeline was created, they were able to share that pipeline to the whole community as a whole pipeline that was very easy to run. Now, the whole reason for them for their existence to some degree is to create science and the results and also methods that they share with the open with the community as a whole. So that their concept of open science. So in that sense, the interoperability that CodeOcean offered by being able to transport this pipeline to another user was very, very strong for them, and they really enjoyed using CodeOcean to further their mission.
All right, and with that, I think we're going to start to wrap things up. So, in summary, we are presenting FAIRR Analytics as an answer to the ongoing reproducibility crisis. The key to this being that all aspects of an experiment need to be tracked, not just the code data and environment, but also the metadata, compute resource, parameters, lineage, provenance, and a whole lot more. And by tracking all of these components, we are able to guarantee reproducibility and optimize for reusability of our computational experiments. And of course, we could not end this without saying that CodeOcean is a centralized, connected system that tracks computational experiments and also facilitates the sharing and collaboration of them. Now, want to thank everyone for attending this webinar. We did have a few questions come in throughout the presentation, so we'll go ahead and answer them now. Just as a reminder, you can still continue to submit questions through the chat. Let's see. Daniel, are you able to see the questions that came in?
Actually, I'm not.
Okay. Maybe Lauren, could you read them to us?
Yes, absolutely. One moment. I'm seeing one question that came in that said, you mentioned Git is used for version control of code. Do you integrate with existing Git repos or like GitHub?
Yeah, so we integrate with GitHub and GitLab and also Bitbucket. So these are the ones, the providers that we currently support, and we might offer more as we go down the road. But that is the most dominant request of our customers, and so that's what we support right now.
We had another question come in asking, have you considered to use research object?
I'm sorry, research.
Just use research object. We can also have another question come in that says, we process large amounts of data on the order of petabytes. Is your system able to handle tracking of data this size?
Right. So CodeOcean doesn't place any limitations in the size of the data that is analyzed. If there are any limitations from your cloud provider, such as AWS, then CodeOcean analysis will be limited by that as well. But that is usually not the case, and definitely not on the petabyte scale. We have customers such as Allen Institute that work with multiple petabytes of data, so definitely that is something we routinely encounter and also support.
Wonderful. And I'm seeing one last question, and that is, once the slice of the database has been made to track the three, how does a change affect tracking? If the code has been changed, is the database aware of it?
So yeah, if there is a change made to any of the components, that will not affect the lineage. That will not affect the provenance of that specific result. So you can make any change you want. It will create a new lineage associated with new versions of the triplet components. So the data, the code, and the environment. So the answer is yeah, we track that, and it doesn't destroy any of the lineage.
Yeah, and just to add to that, we mentioned in the slides that Git is one of the technologies that's used in this case to track changes in code. So Git is used for version control, and that technology is fully integrated into our system. So users are able to use Git to version control their work, and it's incorporated in such a way that users don't even realize that they're using Git. They don't need to have that expertise, but they're still able to take advantage of that technology because it's been integrated into the system. All right, so I think if there are no more questions, we want to thank everyone for attending today. If there are any other questions, please feel free to reach out to us. You'll all receive an email with the recording of today's webinar for your reference. And with that, thanks everyone.
Thank you very much for joining.
Enjoy the rest of your day.